Hi, good afternoon, everyone. So my name is Aaron Hoskins, and I'm here at the University of Wisconsin-Madison. It's my pleasure to welcome you to, I think, the first RNA Collaborative Seminar of 2025. And this is also the first time the seminar has involved two institutions presenting simultaneously. So hear from two very cold universities here in Wisconsin and Alberta, Canada. So it's my pleasure today to introduce Dennis Halterman, who's a scientist with the USDA stationed here at UW-Madison and also affiliated with the Department of Plant Pathology. So with that, take it away, Dennis.
Thanks, Aaron. All right. So today, I'm really honored to be here at this RNA symposium. I feel a little bit like an outsider because I haven't been a part of the RNA Collaborative before, but um, some of my work has always involved working with RNA, and Aaron's trying to change it a little bit more. So I'm a little more involved in RNA biology. I'm really excited about the work that we've got going on together. So today I'm going to talk about the molecular warfare between plants and microbes at the RNA level. And I realize that most of our audience probably isn't very familiar with plant pathology and the interaction between plants and microbes, but I want to take a little bit of time and give a little bit of background before I get into the recent results that we've had. And that work is going to involve some collaborative work we've done with Sumeng Dong at Nanjing Agricultural University. And then I'll kind of lead into some future work that we're working on with Aaron at the University of Wisconsin-Madison. So as Aaron mentioned, my lab, I'm part of the USDA. I'm part of the Agricultural Research Service, which is the research arm of USDA. And I'm located at the University of Wisconsin-Madison Department of Plant Pathology. And the larger picture research that goes on in my lab is to develop potato germplasm and enhance that we can hand off to potato breeders who are in the job of whose job it is to develop new varieties of potato. Specifically, my lab is interested in understanding the molecular basis, the molecular interactions that go on between potato and the microbes in the environment, and understanding a little bit more about how disease resistance works and how pathogens overcome that resistance. So the question I get a lot is, why do you work on potato? Well, my quick answer to that is it's such a fun crop to work with. It's a vitally important food crop worldwide. So it falls only behind wheat and rice as far as a food crop in the world. And it's a critically important industry in Wisconsin. Wisconsin's the third largest producer of potatoes in the US, behind Idaho and Washington State. I like to say we have a respectable genetic and genomic tools for potato. As an outcrossing heterozygous tetraploid, getting genome sequence has been an uphill battle, but we now have genome sequences available for many cultivated potato varieties and pangenome sequence available. And a lot of my work involves studying the wild relatives of potatoes. So we have this incredible genotypic and phenotypic diversity present within the section Patota that we draw on and study a lot to identify important traits for the development of new potato varieties. And that can include, color, shape, flavor, texture, and most interesting to me, plant disease resistance. So a little background on molecular plant pathology. And so people study this and get PhDs in this area, but I'm going to try to sum it up in just a few minutes. Plants get sick from a lot of the same organisms we get sick from: viruses, bacteria, fungi, etc. And the difference between microbes and pathogens is that the pathogens alter the plant to make it a better living environment for themselves. And they do this through using a number of proteins we call effectors that modify the host to suppress innate defenses and ultimately cause disease. On the flip side, plants have evolved ways of recognizing the presence of these effectors. And one of the main ways they do this is through the use of these nucleotide binding leucine-rich repeat resistance proteins that are located in the cytoplasm that recognize the presence of the effectors and then go on to elicit a very strong resistance response that ultimately results in a programmed cell death that we term the hypersensitive response. So in essence, the plant cells sacrifice themselves for the greater good of the leaf or the rest of the plant. So these pathogen effectors are produced by all different plant pathogens. They're encoded by genes in the pathogen, but function within the plant cell or in the apoplastic space just outside the plasma membrane. And pathogens can have hundreds of these effectors with various functions, but ultimately their goal is to manipulate different metabolic processes within the plant to suppress host resistance and cause disease. And as I mentioned on the plant side, we have these resistance proteins that equally, there can be hundreds of these in the plants. Potato, for example, has about 750 putative NLR type resistance genes. And they can detect the presence of these effectors either directly through protein-protein interactions or indirectly by interacting with the targets of these pathogen effectors. But regardless, this interaction results in a conformational change within the proteins that results in the formation of a multimeric, either a pentameric or a tetrameric structure that until only recently we didn't really know existed. But we now know form these structures that form a pore within the plasma membrane that allows calcium to transport and ultimately results in this hypersensitive cell death response. Since this is an RNA group, I want to focus my talk on the role of RNA in plant microbe interactions. And I'm going to break this down into just three sections. The first two are just kind of a short review, small RNAs, plant pathogenic viroids, and then the rest of my talk, I'll talk about the recent work we've done looking at the manipulation of the spliceosome and the role of intron splicing in disease resistance. Small RNAs, many of you are probably familiar with this. MicroRNAs and small interfering RNAs are produced by plants that can target genes within the pathogen. So they go through this process, and I'm not going to go through every step of this, but I just want to point out that we now know that these small RNAs get packaged into secretory vesicles, either through a Golgi-dependent or a Golgi-independent method, where they make their way into the extrahostoreal matrix between the pathogen and the plant, and then are taken up by the pathogen where they can affect the expression of the target RNA in the pathogen. And vice versa, pathogens can produce small RNAs that make their way into the cytoplasm and into the nucleus of the plant cell host, where they can also manipulate the expression of target genes. I point this out because this molecular interaction is a really kind of a different level than the R gene effector interactions that I talked about previously. And it offers some potential control options for plant pathogens that people are exploring now, including host-induced gene silencing, where we can express pathogen targets within the plant cell, producing the double-stranded RNA that makes its way into the pathogen, or we can simply spray the plants with double-stranded RNA, and these small RNAs can be readily taken up by, in this case, fungi, to affect the expression of the target genes in the pathogen. And whether this works or not is kind of waiting to be seen at this point. We know these pathogens can take up small RNAs. The trick is to find the right target that can limit its growth in the wild. But the idea is that we can have another mode of action in addition to fungicide and other cultural changes that we can make to control diseases. I want to take a moment to a shout out to plant pathogenic viroids. Many of you may not be familiar with this class of plant pathogens because it is specific for for plants. They are not found in animals. There have been some that have been found in fungi and oomycetes, but they are primarily specific for plants. And if you boil down a pathogen to its most simplistic state, that's what you get in a viroid. These are naked RNA. They're covalently closed circular RNA genomes. They encode no proteins, so they rely on the host machinery to replicate and move throughout the plant. And it can be very small. So as small as 246 nucleotides in total, up to about maybe 550 or 600 nucleotides. The first viroid was identified back over 50 years ago. And that was potato spindle tuber viroid that was identified and cloned and by someone in the US Department of Agriculture. And they have conserved domains within the viroids. They also have these pathogenic domains that are variable. And the idea is that these pathogenic domains induce RNA silencing through the production of double-stranded RNA and small RNAs. And these viroids are quite numerous. There was a paper published just a couple of years ago in Cell where they mined transcriptome and metatranscriptome data and found over 11,000 covalently closed circular RNAs that are viroid-like. And out of this 11,000, only about 1,200 of them had similarity to known viroids. So there's this vast unexplored area of research relating to plant pathogenic viroids, what they're doing, how they're moving, how they're causing disease. And we still don't really know how they originated. The hypothesis now is that they were probably at one point a helper virus or a satellite virus that evolved to be able to replicate and exist on its own. I'm going to switch gears now into talking about the manipulation of the spliceosome and intron splicing. And I want to preface the rest of my talk with this table that points out we've known that intron splicing has been an important component of plant disease resistance for decades. This is just a table of disease resistance genes that have alternatively spliced introns. And in here is actually some work that I did as a postdoc at Iowa State University in looking at barley powdery mildew resistance. We know that a lot of these resistance genes, these NLR genes, contain introns that can be alternatively spliced. What the function of them and what the role of this alternative splicing was, we don't really have a great idea, but some of the work we've done recently helps to shed a little bit of light on that. So I want to talk about some of the cast members in this work. We're going to talk about potato late blight. It's caused by an oomycete pathogen, Phytophthora infestans. It was involved in the Irish potato famine 170 years ago, and it's still a big problem today. It's very well known as a plant pathogen that's able to overcome resistance that we deploy into the field. And it does that using this toolbox of up to 500 different effectors that function to manipulate the host, either potato, tomato, or tobacco. And on the host side, we have our cast member called RB. It's a resistance gene, an NLR type resistance gene that was initially cloned from the wild potato species Solanum bulbocastanum, and that's where it got its name, resistance from bulbocastanum. And it recognizes the presence of a single effector called IPIO1 from Phytophthora infestans. And all of the Phytophthora infestans strains that we've sequenced all have IPIO1 present in them. And so we can introduce this RB gene into potato. This is a susceptible potato on the left, and then the plant that contains RB in the middle. And you can see this dramatic difference. The single resistance gene has a big effect on that. And the USDA scientist, my predecessor, John Helgeson, was involved in cloning and working on this RB gene. So I've kind of carried the torch with trying to understand how this RB gene works. In trying to understand how IPIO functions within the cell. So I mentioned that Phytophthora has 500 or so effectors. There's been a lot of work that's been gone on over the past couple decades to try to figure out what all of these effectors do. We used a yeast-2 hybrid system to look at proteins in the plant that are interacting with IPI-01, and we had a long list. This is just kind of 10 of them that were most interesting. And I want to point out one of these, this Phytophthora IPI-01 interacting protein 6, which is a pre-mRNA splicing factor called CWC15 that's involved in the spliceosome. So at this point, we were kind of working through all of these genes, trying to figure out what they might be doing. We didn't really have any knowledge about the role of any of these in disease resistance. Then I went to a meeting, the Congress of the International Society for Molecular Plant Microbe Interactions, and my now collaborator, Sumang Dong, gave a talk at the meeting where on work that they had done to look at Phytophthora effectors that manipulate intron splicing in this case, tomato. And so they used RNA-seq to look at alternative splicing globally within the plant in following infection with Phytophthora infestans. And overall, they didn't see a huge increase or decrease in alternative splicing overall, but there were some genes specifically that were induced or repressed in their intron retention. And one of this receptor-like protein kinase he used to develop a luciferase assay where they could look at whether or not the intron is spliced. In this case, if the intron is spliced, you get luciferase activity. If the intron is retained, you get a premature termination codon that leads to a non-functional protein and no luciferase activity. So they used this reporter and tested it in combination with 80 or 90 different Phytophthora effectors and found 9 of them that significantly reduced the splicing of the intron of this reporter. And one of these happened to be IPI-01, my favorite effector that we have been working with. So I was sitting in the audience listening to him give this talk, and these fireworks went off in my head. We talked later at the meeting and decided that he's got this effector that's affecting splicing. I actually have a protein that is a component of the spliceosome that's interacting with the same effector. Let's try to study this a little bit more and figure out what's going on. So that led to the work that I'm going to talk about now that we published just recently in the plant cell. So we looked first at the RB gene. So it's the resistance gene in this pathosystem. We know that resistance genes can be alternatively spliced, and so it wasn't a big surprise when we look at the intron within RB. There's one intron present, and it is alternatively spliced. So this is RNA-seq data, and we find both variants, and this intron splicing leads to 2 different transcripts, one where the intron is retained, then that leads to a premature termination codon that is a non-functional protein. But if the intron is spliced, that leads to a functional protein that, as I mentioned, can go on to recognize the presence of IPI-01 and turn on this hypersensitive resistance response. And this is just an example of various potato plants that contain RB, where we can see both transcript variants present under normal growth. This is another wild species that has an RB homolog from stoloniferum, and this is PT29, which is Solanum bulbocastanum. All 3 of these have RB. This is tobacco that's expressing RB. In all of these cases, we can see that RB is alternatively spliced. Then we went on to show that RB overexpression stresses out the plant. So this is something, a hypothesis that people have had and have shown in a couple of cases where you overexpress resistance proteins in plants and it can stress them out. It's no surprise given that these proteins are cell death proteins. They lead to this hypersensitive resistance response, and if they are inappropriately activated, they could lead to this microscopic cell death, but globally that could reduce the the metabolism of the plant and stress them out. So in this case, these are tissue culture plantlets of wild type potato cultivar called Favorita. And when we just express the wild type RB, we don't see any effect on plant metabolism or fitness. But if we purposely remove the intron and only express an intronless RB, so this is only making the coding sequence and it's overexpressed, we can see that that overexpression leads to a reduction in plant size, both stem height and root length. But if we permanently remove that intron, so in this case, we can change the nucleotides at the splice sites, both 5' and 3', we can see that in that case, the plant fitness is okay. And this is reflected below ground as well. This is the wild type tubers from that plant, and this is the plant that's overexpressing the RB, the functional RB variant. And you can see that this is not a zoomed out picture. These are the smaller size tubers. And what's interesting is that the number of tubers wasn't significantly changed. It was just the size of the tubers that was impacted by the overexpression of the RB protein. Then we went on to show that the RB splicing is specifically induced by Phytophthora infestans. So this is a graph showing the splicing ratio of the intron spliced coding sequence in the intron retained IR version. And you can see during P. infestans infection, you can see this ratio go up. So the splicing is increased during infection, but it is not during infection with another Phytophthora capsicum. And we used multiple strains of P. infestans, and we saw a similar thing where the ratio of the spliced version goes up 2 or 3-fold during infection, but not with the water control and not with capsicum infection. Then Sumeng and Biying made a reporter similar to the one they used before, but in this case, they replaced the receptor light protein kinase with RB. And in this case, if the intron is retained, we get no activity because it makes this premature termination codon and a truncated protein. But if the intron is spliced out, we get luciferase activity. And then they went on to test these various splicing regulatory effectors and found that the only one that increased luciferase activity or intron splicing was IPI-01. And then this is the control where there's no intron and everything is producing luciferase as we would expect. We already had preliminary data that IPI-01 interacted with the CWC15 protein, this spliceosome component, but we went on to show that again using a different yeast 2 hybrid system and also in pull-down assays. And I want to point out that in potato and tobacco, there are actually 2 CWC15 genes present on different chromosomes. We haven't been able to separate any functional difference between these 2 different genes, but why they're on different chromosomes and not just a duplication locally, we aren't really sure what the role of that is, but they seem to function very similarly. They both interact with CWC15 and you can knock them both down and they both have similar phenotypes. We went on to show then that the resistance, this RB resistance to Phytophthora infestans is dependent on the spliceosome. We used RNA interference to knock down the expression of a couple of genes that are important in spliceosome assembly and function. And they basically, when we knocked those out, made the plant susceptible. So these are plants with the RB gene. If you knock out the spliceosome, you knock out resistance conferred by RB. If you knock out this splicing associated gene GRP7 that's not in the spliceosome, we don't see that same effect. So it seems to be a spliceosome specific effect that is impacting resistance. And this is just looking at, again, resistance lesion size and its splicing ratio of RB, where we can see that we knock out these genes, we knock out the splicing of the intron. We went on to use PLA-B to show something very similar. This is an inhibitor of spliceosome activity, and at high levels, this can knock out the hypersensitive resistance response that we see mediated by RB, and also affects the splicing activity of RB. We did some work, confocal microscopy, to show that this interaction takes place within the nucleolus and the nuclear speckles within the nucleus. I won't go through all of this, but the nucleolus and the nuclear speckles are associated with intron splicing in plants. And so it was no surprise that these IPIO1 and CWC15 proteins translocate there together. And during this interaction, we see this induction of nuclear speckles and nucleolus localization. And so our working model then is that under normal growth, the intron of RB is not spliced. And the plant does that because it doesn't want to stress itself out. But it wants to keep RB around because it's recognizing the presence of this essential effector within Phytophthora. And so it's come up with this elegant way of preserving its activity by saying, okay, well, we're going to stick an intron on RB. It's not going to be expressed during normal growth, but we want that intron to get spliced and make the RB protein when Phytophthora is present. So we think that this intron evolved as this kind of a sensor of the activity of IPIO within the plant cell. IPIO1 is functionally, it's not there to splice the intron of RB because it wouldn't want to do that. So ultimately this work doesn't elucidate what IPIO1 is doing or what its target is globally, but it sheds some light on, gives us an avenue of research to carry on. And so we're doing some work now. This is just a simple experiment. Can we duplicate the intron within RB and show that it's still functional? And lo and behold, we can copy paste that intron within RB, and we can see that both of these introns work similarly under, alone. Both of them are not spliced, but when we add IPIO1, we can see that both of them are spliced in the plant. We've been looking at trying to find other targets, other genes that are alternatively spliced in the presence of IPIO1. So we have these transgenic plants, and we're using RNA-seq and intron retention finder to try to find genes that are alternatively spliced in the presence of IPIO1. And surprisingly, we have a very small number of targets at this point that we're following up on. And then other work we're doing in collaboration with Aaron at UW Biochemistry to try to understand the role of CWC15 in splicing. So we're using mutagenesis and looking at interactions in RNAi of CWC15 to try to figure out what its role is in splicing and disease resistance. And with that, I think my time is about up, so I will stop and take any questions that people might have.
Great. Thanks, Dennis. And so we have time for a couple questions. So if you have a question, type it in the chat, and then I'll read it out loud to everyone.
So one question in there is there a polyadenylation signal within the retained intron? It's a good question. I'm not sure that we've looked at that. We know that intron retention can induce an RNA degradation pathway that leads to degradation of the RNA. We've looked at that, and we can still find the truncated RNA in the plant quite readily. So it doesn't seem to be degraded through that system. Whether that is dependent on polyadenylation, I'm not sure that we've looked at that specifically, but we should be able to in our RNA-seq data. I'd have to look at whether that was poly-T amplified or whether it was random primer amplified to see that. So that's a good question. Yeah, I don't know the answer, unfortunately.
So maybe we have time for one more question before we turn things over.
I'll stick around, so if people throw things in the Q&A, I'll keep an eye on it and try to answer.
All right, let's see. A little bit. All right, I'm not seeing anything else come through. So why don't we turn things over to Alberta? Thank you, Dennis.
Thanks.
Thank you. Yes, it's also cold here in Lethbridge, Alberta. So we're part of the Alberta RNA Research and Training Institute. And I'd like to just invite Mark Lee, a master's student in Dr. Stacy Wetmore's lab, to give his presentation.
All right. Thank you, Laura, for the introduction. Um, yes, I'm Mark Lee. I'm from the Wetmore group at the RT RNA Center in Lethbridge. And today I'm going to share my research as a second-year master's student on the role of messenger RNA modifications in ribosomal decoding. The Wetmore group is a computational lab, so this is a molecular dynamic simulation study. So as we know, RNAs are responsible for actually building proteins in the cell. Um, the process of protein synthesis is controlled by collaborating between tRNA, mRNA, and the ribosomal RNA. And essentially, a tRNA will attempt to bind into the A site and make base pairing interactions with the messenger RNA. And these three base pairing interactions are known as the codon to anticodon mini helix, and this process is known as decoding. We also know that RNA is chemically modified and that these modifications influence the chemistry and the functioning of RNA molecules. And they can range from smaller changes like isomerizations to much larger, more complex modifications. And actually, ribosomal RNA and transfer RNA have very abundant modifications and very diverse chemical structures. And they've received a lot of attention in the research and thus have a lot of established roles. But on the other hand, messenger RNA modifications, especially those occurring in the coding region, have less well understood roles. And this is despite the fact that we know they have very good potential for preventative and therapeutic applications. One example is the N1 methyl pseudouridine modification, which was substituted in all of the uridine positions in the COVID-19 vaccine and is widely considered one of the reasons why it was such an effective vaccine. So if we want to understand how to better use and exploit the properties that mRNA modifications allow for, it's important to understand their roles in fundamental processes like decoding. And so for my work, I've been collaborating with two different experimental labs. The first is the Wetmore group, who runs experiments using in vitro reconstituted translation systems, and they measure how fast peptide bonds are formed and the influence of mRNA modifications on this process. The Dunham group is also working on developing structural snapshots during the translation process with different modified mRNAs. In the Wetmore lab, obviously, we are performing simulations to better understand the atomic motion and structural dynamics that modifications cause changes for decoding. So first, we're going to look at some of the experimental data, just for some of the background. The Wetmore lab has introduced the inosine modification into each of the three positions of the codon. And they measured how this influenced the rate constant of peptide bond formation. So when they added inosine into the first position of the codon, they found that there was a 10 times slow down of the rate constant for peptide formation. But when it was added in the third or second positions, there was no significant influence. So in order to rationalize this, we wanted to perform simulations with the same corresponding systems. And there have been previous studies that use MD simulations to study decoding, but they use this very tiny portion of the ribosome. The reason why we use a truncated region of the ribosome is because it's such a large biomolecule that it becomes very difficult to simulate and get results because of how much computational resources it takes. However, this region doesn't actually let us see what the impacts are of decoding. For example, if a tRNA were to be dissociated from the mRNA, we can't really tell if this is just an artifact of the model itself being too small or something else. So for our work, we're using a larger model with the entire tRNA included, and a larger portion of the A site included with the magnesium ions and ribosomal proteins in RNA. And we're also including a simulated physiological water box. And we're running 4 replicas of 1 microsecond simulations. And this is about 10 to 100 times more sampling that has been done for studies on decoding, especially with modifications. So now that we have all this simulation data, it's important to know what we actually want to measure to understand how it impacts decoding. And so what we looked at first was how the ribosome is actually involved in this process. And the ribosome is involved in a process called monitoring, where different parts of the ribosomal RNA will contact the minor groove of the mini helix region, and they push waters out of that region to stabilize the optimal alignment of the mRNA and tRNA. We'll also look at the mini helix structure itself by looking at the distance between each base pair and their alignment, and the conformational dynamics of the tRNA and mRNA. So first, we're going to look at the helix 44 monitoring adenines, A1492 and 1493. And you can see that these are flipped out of their helix and they contact the minor groove region. And they can actually flip back in their helix. And one way that we can measure this is through the on or off states of the monitoring angle shown here. So this picture is showing an on state where both adenines are actually flipped out to contact the minor groove, but you can see that we can have this green state on the left where both are flipped out, but there's also a state where one is flipped in and one is flipped out, and then where both are disengaged in the red colored state. So first I wanted to look at the experimental structures. So I plotted the monitoring angles where the x-axis is 1492 and the y-axis is 1493. And we noticed that there was a significant portion in the on-on state, some in the off-on state, and some in the off-off state, and intermediates between these states as well. And when we compare this with the simulation data, which is shown as a free energy landscape, where the darker blue areas are lower energy states and the red is higher energy states, we can see that for the second position inosine modification from CGU to CIU, there wasn't a significant impact in changes in this energy landscape. But for GUG, we can see that GUI differs, but also in IUG, we see that this is the only model where the off-off state was truly sampled for a significant portion. So this indicates that both adenines are actually being flipped back into their helix. So also to look at G530, the other monitoring base mentioned, we can also use these colors to explain them. There's the on, semi-on, and off state. For the CGU and CIU, once again, we see there was little change in the dominant conformation observed, and GUG was spent most of its time in the on state, whereas GUI was in the semi-on. But once again, for IUG, we can see that the G530 base spent nearly half of the simulation time in the off state, where it was disengaged. So now we know that all 3 monitoring bases are not helping to stabilize the mini helix. So we wanted to look at the mini helix structure itself. And to look at this, we measured the distance between the C1 prime atoms of the mRNA and tRNA for each of the 3 positions of the mini helix. And you can see also we measured the planarity between the opposing nucleobases as the planarity between the rings. And you can see that IUG has broadened distributions in all 3 positions compared to other models. The GUI in the 3rd position also has a very broad distribution, but you can see that it didn't actually influence the 1st or 2nd base pairs, and it didn't influence the distance between the C1 prime atoms, which means that it likely wasn't actually pulling apart the mini helix. So we next looked at the actual conformations of the tRNA and mRNA sampled across the simulations. And we found that there were 3 conformations, the stacked, wobble, and 5' disrupted, where the overall mini helix remains intact with some flexibility at the wobble position or the 5' end. But there was a 4th conformation where the entire mini helix was perturbed, which we've denoted rejected. And if we look at each of the systems studied, we can see that IUG has nearly half of its simulation time in the rejected state. So this helps to rationalize why the IUG mRNA codon might be causing a cognate tRNA to be rejected. So if we look at the dominant trajectories for each of the systems, we can see that for GUG or the non-IUG models, the mini helix remains intact and the orange monitoring bases remain in the minor groove region. However, for IUG, we can see that the orange bases are completely disengaging and the tRNA is being rejected by dissociating from the mRNA. So now that we've established this with the inosine modification, we also wanted to compare with the M1G modification with the COTMA lab results. And they found that the first and second position M1G caused a significant slowdown of translation or dipeptide formation, whereas the third position had negligible impact. And comparing with our conformational distributions once again, we can see that the first and second position M1Gs caused very high rejections, whereas the GU M1G in the third position did not. So we can see that by changing the hydrogen bonding patterns in the models, we can provide a rationale for the experiments that aligns very well with the kinetic data. And for future directions, we want to study a wide variety of different modifications, whether they're incorporated by enzymes, they're the result of damage, or synthetic modifications, especially those that don't have experimental data already, because we've established that the model can be very predictive. So with that, I'd like to thank my supervisor, Dr. Stacy Wetmore, and my collaborators from the CUTMO and Dunham labs and the Wetmore group. And thank you everyone for listening to my presentation.
Awesome, thank you, Mark. If anybody has any questions, please type them in the Q&A. I have a question. When you're doing your modeling for that mRNA mini loop, it was only how long was it? 3 nucleotides?
Yes.
What do you think you're going to see anything different if you model like a longer stretch of nucleotides?
So since the anticodon is usually a fixed length, it doesn't really matter if we included more because they would still be like discrete binding sites for each of the tRNAs. Like the P site tRNA, for example, binds to the AUG codon, and then the A site tRNA binds to the next 3 nucleotides. There are some cases where you can have recoding where there's like elongated codons of 4 nucleotides, but we didn't study that specifically.
Okay, so you don't think there would be much different?
Oh, there could be lots of differences, but that wasn't like the focus of this work. That would be an interesting thing to look at, though.
Okay, awesome. If you do have any questions for Mark, stick around and you can put them in the chat and he can answer them there. And I guess we'll move on to our next speaker. Dr. Jessica Willey is a new professor in the Department of Chemistry and Biochemistry here at the University of Lethbridge. And yeah, I'll take it away.
All right. Well, thank you so much for having me and giving me the chance to present some of our latest research. And today I want to pick up where Mark kind of left off with the ribosome and how it could help unlock more efficient biotech. The biotech industry, as you may know, is $1 billion industry. And when you're thinking about biotech products, you may think of products like insulin, vaccines, or enzymes that help your laundry get clean at lower temperature, or life-saving drugs such as antibodies. Now, what these all have in common, of course, is that are protein products, which we will typically industrially produce in these big vats, these bioreactors. In the lab here, we are on a little more modest scale, but our real workhorse here is recombinant E. coli that has been transfected with some sort of genetic information that allows it to produce these valuable products. Whether you work with recombinant E. coli or some sort of version of a cell-free expression system, it is not really this type of technology that produces these proteins. It is, of course, the star of the show, the ribosome, that is the actual machinery producing proteins from the genetic information, the DNA and mRNA. And so I like to think of the ribosome as really nature's 3D printer that is taking this genetic information and turning it into a protein. This is happening amazingly fast in the bacterial ribosome. Elongates by about 20 amino acids per second, linking them together into a chain. This is extremely accurate. The information encoded on the codons is translated into proteins with very few errors. And of course, these adapter molecules permit encodability, which means that no matter how many proteins, how many different messages you want to produce, you can do this all with the same ribosome in the background, as Mark has illustrated. And I want to highlight two different things here, is that this amazing 3D printing function is actually encoded and catalyzed by the ribosomal RNA itself, which means the ribosome is a ribozyme. It's an enzyme that does what it does with the help of RNA. And also that this is a massive, very dynamic complex. So if we're just thinking about the smallest ribosome that we work with in E. coli prokaryotes, we are still talking about a 2.7 megadalton complex that is flitting around in the cell doing its job. So if we're thinking about how we can kind of hack the ribosome or alter it. If we want to alter it, we would go for the RNA, which of course is what gives it its function. And this is kind of what my lab is all about, is expanding and modulating the function of the ribosome by modifying its RNA. So we're going after things like the genetic code itself, the active site to expand what it can do, but also going for efficiency. So especially with biotech in mind, some of our engineering principles are to make more protein per ribosome, so to make this whole system more efficient. But also on the flip side, we could think about how we could produce the same amount of protein by using less ribosome. So those are my two kind of topics for today, and I want to start with making more protein per ribosome. Ribosomes are not all created the same. So especially if we look at E. coli, we see on the genome, we have actually 7 different copies of the ribosomal RNA. Here you can see the 16S, the 23S, and the 5S. And every little tick here on this graphic is a small mutation or heterogeneity that is a little bit different between each one of these operons. So as the cell is growing, it is expressing all 7 of these variants at the same time. So there's kind of a pool, a mixture of different ribosomes. And we were wondering, do these ribosomes have different functions? So we built the ribosomes either in vitro, or we purified the ribosomes after they had already been built in vivo. And as you can appreciate, each of these ribosomes here on the bottom, denoted by different letters, actually has a different amount of protein synthesis capability. So these are active to different degrees. And this is especially striking if you compare it to the wild type here. So this MG1655 is the wild type E. coli cell, and it is producing this mixture of 7 different operons. And you can see this is quite a bit less efficient than if you just produce one ribosome at a time. So some of these ribosomes, such as ribosome type A, has a bit of an A-type personality and is an overachiever, so it produces way more protein. So with this in mind, we were thinking, what if we produced a strain of E. coli that only produces one type of ribosome? Specifically this really overachieving ribosome. So this is exactly what we did, knocking out the endogenous sites of the operons and replacing the genetic background with just ribosomal RNA encoded on a plasmid. So this would result in a normalized RNA pool or a ribosomal RNA pool. So all of these would be genetically identical. We're losing diversity, but indeed we are actually gaining protein expression. So here is a panel of a bunch of different bioindustrially relevant proteins. You may recognize something like Cas9, but also CRIM, that is used for producing vaccines, and some metabolics, enzymes on the other hand. And almost all of these experience a boost in protein level expression when we grow them with just the A ribosomes in mind. So this was published last year, and we're hoping that this may get picked up by the bio industry with our patent, so that we can boost protein production at really no extra cost by just altering the ribosome diversity. So with this, back to our kind of engineering goals, we managed to make more protein per ribosome. But what if we could have smaller ribosomes instead and save a little bit of energy that way? So what do I mean by this massive investment? The ribosome, as I said, is a massive complex, and it is very large, and we have a lot of ribosomes in every cell. Even in moderate growth, these are tens of thousands of ribosomes that have to be produced by E. coli. Up to half of its dry mass is just pure ribosomes, and the ribosomal RNA makes up the bulk of total RNA in E. coli. And of course, some of these ribosomes are needed to make some of the ribosomal proteins that kind of decorate its outside and make it more stable. So if we could shrink down the ribosome a little bit, we could save a lot of this resources and energy that could be instead be put into more protein production that is desirable to us. So shrinking the ribosome is a little bit of a large task. So we looked to evolution as an inspiration. If we think about how the ribosome evolved, there are several theories. And one of these theories is through expansion from an ancestral ribosome RNA world to the ribosome we know and love today by expanding slowly its RNA content. And so with this model in mind, I wanted to use this as an inspiration to grow back the ribosome to a more ancestral, smaller state, and seeing if this could still be able to produce proteins, but being a smaller ribosome all the same. So naively, I grabbed my molecular scissors and started snipping away. But of course, the ribosome initially was not very happy with this, and it produced a lot of non-functional ribosomes that would not make any protein whatsoever. So I had to choose a different avenue. And again, it is ribosomal evolution to the rescue. So this is an in vitro evolution platform in which I randomize a bunch of different ribosomal RNA structures, try to build ribosomes out of them. And then if they are active, we can pull them down and enrich only the active ribosomal subvariants. So it's a little bit of a twist on ribosome display, but it means we can screen thousands of different ribosomal variants in one single reaction. We don't have to know everything about the design in order to get successful variants. And then those winners can be combined back again in order to keep shrinking down the ribosome to its most essential components. So one example of how this could look like is if we want to remove this little helix right here in order to make a smaller ribosome. Of course, this is a very modest minimization, but it was challenging nonetheless. We would snip this away and replace it with a little linker. And this linker can now be evolved for optimal folding. So if we compare our initial linker, it is very much dead. That is what I designed naively with my molecular scissors. Compared to the wild type ribosome, it is almost dead. But as we're going through iterations of evolution, after several rounds of this evolution procedure, the library has actually gained back some protein production capability. And if we grab this and do some NGS sequencing, we can actually see what are the linkers in this region that are preferred by the ribosome in order to produce active subvariants. Now, we didn't just do this with one specific little deletion here. We did this with many other ones. So this is kind of all the data. So there's a lot on this slide, but I will note that right here in the top left corner, we have the wild type ribosome. So anything that is lighter than this color is going to be less protein translation, and anything that is darker than this color is going to be more protein translation. And so we have all of these numbers denote different deletions, different minimal ribosomes that we produced. And as you can see, even though some of the initial linkers are appearing to be quite dead, a lot of them can actually be rescued by our evolution approach and regain some of the protein synthesis capability and even exceed how much protein they make compared to the wild type ribosome. We typically do this with an SFGFP reporter, but I also tried this with a mini helix, which this is kind of to scale, much, much smaller of a reporter compared to GFP. So it's not that hard of a job compared to GFP to make this little mini helix, which can be as small as 12 amino acids. And you can appreciate that the mini ribosomes also really like to make mini helixes, and quite a few of them are way more active than the wild type ribosome. This, by the way, this mini helix, if you have some use for them, this is published, so you might want to use that in some of your own assays. And we can drive this even further. So we can start combining these minimizations, these deletions together into something that looks nothing like the modern ribosome that we know and love today. So this is one combination of the small subunit deletions, and it lacks up to 43% of its original RNA. And so I marked this out here. Anything that is in gray is what is left. Anything that is in purple is what we have cut away. And you may notice some proteins here marked out in magenta, and these are actually ribosomal proteins that may be missing in action because we've deleted away all of the ribosomal RNA that they would need to interact with the ribosome. So these, we are currently working in confirming that these are completely gone via mass spec. And you can also see in this RT-PCR gel, compared to the wild type ribosome, this deletion is markedly, visibly, much, much smaller in the ribosomal RNA. So with this, I hope I could convince you with my talk that sometimes, even with the ribosome, less can be more, whether it is diversity or the ribosomal RNA itself, if you engineer it properly. And I can show you that ribosomes can be engineered to be more efficient in protein production for biotechnology, but also to be smaller. And with this, I want to acknowledge the new members of my group, especially Camilla, who has helped in some of the first story, and my funding agencies. And if you'd like to know more, please check out my website.
Awesome. That was so good. Okay, we have quite a few questions actually. So, is there a fitness cost to having only A ribosomes in E. coli?
So as far as we see for the protein production, it obviously boosts this specific protein production, but it may be that under certain stress conditions, these cells do more poorly because they are only running on this one ribosome. So of course, if we're growing them under optimal conditions in the lab, these cells have no stress whatsoever. They are being pampered. So that should not necessarily be a drawback for them.
Awesome. Okay. Are the more efficient ribosomes more error prone?
We actually haven't checked error proneness from those specific ribosomes. However, in just from our experience, when we produce enzymes, for example, that have to have a function, they seem to be just as active as enzymes produced from a mixture of different ribosomes. So from functionality standpoint, there isn't anything too obvious, but we might have to do some specific error type assays to really nail that down.
Okay, well, they're coming in hot and fast. Okay, why are the various ribosomes in the cell? Is there any evolutionary selection for productive ones?
Oh, I thought the same thing, right? If some of these ribosomes are lemons, why keep them around at all? But my working hypothesis is that some of these ribosomes, they may be kind of duds if everything's going well, But under certain stress conditions, they have secret superpowers that are activated and may help the cell survive like low nutrient conditions or maybe even antibiotic conditions. And actually, my student here, Ella, standing in the middle, she is holding some E. coli's that are expressing only certain ribosomes, and she is testing whether they have more or less sensitivity to antibiotics. So we're testing that as we speak.
Awesome. How to explain the difference in activity for the different ribosomes A, B, and C, etc.
So the differences between them on a sequence level are really, really minute. So it really comes down to small changes in the dynamics of the ribosomal RNA during translation. So this is really fine-tuning the translation. That's how I imagine it.
Okay. Okay. Do we have the sequences of the ancient ribosomes? If so, how are those performing for protein synthesis?
So the ancient ribosome model, it is all based on phylogenetic data because we don't have like actual fossils of these ribozymes. So this is all theoretical, right? And I'm actually the first person to go and try to construct these ancient ribosomes based on the model and seeing if they have activity or no activity at all. So I guess this is the first thing we know so far.
Do you think that this will apply also to mammalian cells?
Ooh, that's very difficult to say because mammalian ribosomes are crazy, right? They've kind of taken this expansion evolution that I was showing you from the ribosomal world, the RNA world to the prokaryotes, and they've taken it even further. So their ribosomes have all sorts of weird tentacles and decorations on the surface that may be needed for regulatory processes or optimizing error rates. But I don't think I dare to touch these because I think it would break them immediately.
Okay, last question. From evolutionary perspective, can we say the expansion of ribosome is the result of selection to produce larger proteins as mini ribosomes are better at synthesizing the small proteins?
That is a very cool hypothesis, maybe. I don't actually know if larger proteins necessarily come later in evolution, but that might be an explanation of why the ribosome had to grow with its challenges, if you will.
Awesome. Okay, thank you so much, everyone, for presenting and for attending today. I just want to shout out the next RNA Collaborative Seminar is on March 12th at 10 a.m. Eastern Time. So thank you, everyone, and thank you, speakers. It was great. Have a great rest of your Wednesday.